For our experiment, we decided to test how different bacteria are affected by different treatments. The first thing we had to figure out was how exactly bacteria cells are different than human cells. Bacteria cells are prokaryotic, making them different than human cells, because human cells are eukaryotic. The first thing we had to do to set up this experiment was fill 6 petri dishes with agar. Agar is a gelatinous substance derived from seaweed. The gelling agent is an unbranched polysaccharide obtained from the cell walls of some species of red algae. The next thing we had to do was clearly label which dishes would be used for which of our treatments. We took spit from Nick Wombat Borelli's mouth and put it on each of the agar dishes. We used 3 different treatments. We used penicillin.
Oh, Sandy was like a big bear.
And honey. And wombat's vinegar. The first antibiotic was discovered by Alexander Fleming. It was discovered on September 3rd, 1928. Fleming was cleaning up a messy lab when he found plates that had been coated with Staphylococcus bacteria as part of another experiment. One of the plates had mold on it. The mold was in the shape of a circle, and the area around the circle was free of bacteria. This mold was penicillin and became the first antibiotic. Penicillin works by inhibiting the formation of peptide crosslinks in the bacterial cell wall. This antibiotic causes cytosis, which is death due to osmotic pressure. One of the problems with antibiotics is that some bacteria can acquire resistance. Acquired resistance is when an organism can withstand the effects of antibiotics. Antibiotic resistance evolves via natural selection, acting upon random mutation, but is also engineered by applying an evolutionary stress on the population. New antibiotics are developed from plants or other different ways, especially from the rainforest. Although antibiotics are generally safe, they are associated with a wide range of adverse effects. Some possible adverse effects range from fever, nausea, to diarrhea. As you can see by this petri dish, vinegar was our most effective treatment. The kill zone was biggest in vinegar compared to honey, penicillin, and the control. Here is our graph of the data. As you can see, on the x-axis is our treatment type, and on the y-axis is the kill zone in centimeters. Our most effective treatment was vinegar, followed by honey, then the control, and then penicillin.